Now I'd like to go back to environmental microbes and tell you a little bit about what my lab is doing. So my lab studies how human intervention affects microbes in the environment, how the human world affects the microbial world. And if you look at ecosystems, for example, a lake or soil, or even a wastewater plant, all of these ecosystems have microbes that help them function properly. And when we perturb them, when we shake them a little bit, for example, if we fertilize the fields around the lake, that can cause an ecosystem to collapse. For example, in the lake, it can cause a harmful bloom that can be toxic to people who live around the lake. In soil, if we add intensive farming like tillage or fertilization, that can cause the soil to degrade and not be able to grow any more food. And in wastewater plants, if we have chemicals in the sewage, that can cause the wastewater plant to fail, and that can cause pollution of, here, for example, the Mediterranean Sea. And what we're studying in our lab is all of these are local effects. We're actually asking if there's something global that humanity does to the microbial world. And our intuition comes from another ecosystem. You know, when I was a kid, we used to go on trips on weekends. And once we got to where we were going, the windshield was covered with dead bugs. And the scary thing is that it doesn't happen anymore, and we can see it in the data. So what you're seeing here is the numbers of bugs, not on windshields, but collected in the 1980s compared to the ones collected 10 years ago. And the insect population has dropped 80 to 90% worldwide. And these insects are important. They pollinate, they help us create food. But I would argue that microbes are much more important. And you can take it, if you don't believe me, you can take it from Louis Pasteur, who said that life would not longer remain possible in the absence of microbes. And this is a guy who dedicated his life to destroying microbes through pasteurization. But not all is bleak. Microbes also present opportunities for discovery, this response of microbes to human life. For example, in natural products like antibiotics or probiotics, in crop enhancements to make crops grow better, in better animal farming, more sustainable animal farming, and in waste management. And I'm going to give you a small example of waste management. In 2016, researchers from Japan found this microbe. And this microbe produces an enzyme called PETase. Now, this enzyme can metabolize the plastic in plastic bottles. Now, this is amazing for a number of reasons. It's amazing because plastic's only been here for like 60 years, and microbes already evolved it as an environment. And the other thing is that this introduces a brand new way to do recycling using microbes. Now, if we want to discover more of these, we have to face a few challenges. And I'm going to talk about these challenges a little bit right now and how we kind of deal with them. The first challenge is sampling throughput. What does it mean? It means that we want to sample more. We want to go into all of these weird environments and look for the microbes there and what they're producing. And the cost of sequencing a genome or a metagenome, a microbiome, has dropped about 100,000-fold since the turn of the century. But we still have to do all the tedious work of preparing the DNA and extracting it and processing it and all of that. And we want to do more for less of this thing. So we're going to use robotics, and we're setting up a new core service at Weizmann called the Environmental Omics Core. And this Environmental Omics Core takes samples from scientists, it processes them, and then it passes them on to the relevant cores, the genomics, the metabolomics, the proteomics, and through analysis, it goes back to the scientist and gives them insights into this new environment. And this is a huge enabler because it enables scientists who are not microbiome scientists to do this type of research exactly. The next thing is dealing with unknowns. Remember I told you that there's like 10 billion to 1 trillion species of microbes? So most of them are unknown. And what you see here is a graph. On the x-axis is the number of samples collected. On the y-axis is the amount of new information, specifically here, the number of new genes that we can find. And we're seeing that in the human gut, with every new sample collected, we're not getting much more new information. But in the soil and marine environments, with each sample, we get millions of new genes that we don't know the function of. And some of them could be the next prebiotic or the next antibiotic or something like that. And so how do we deal with that? we use AI, and we develop new tools to address this problem exactly, and we're taking a page from the Netflix book. Has anyone watched Netflix? So when I was new to Netflix about 8 years ago, they told me I should watch Die Hard. And I was like, okay, that's spot on because I love Bruce Willis. And how did they know? So they knew because they create this huge network of people and content. And for each person, they have their income, they have their cookies in the browser, they have their geography, the age, everything. And for the content, they have the same thing. They have a network of the genre, the cast, the director, the year it went out, and so on. And they know who watched which show. This is a tongue twister, so I have to be aware. So they know which people watch which content. And when a new person comes out, they go to the database, they look at the income, they look at everything, and they match things that match people who are similar to them. This is called link prediction. And we're doing the exact same thing, except not with people and content, but with enzymes that we found in the environment and with pollutants. And this is done in collaboration with the Mohammed bin Zayed University of AI in Abu Dhabi. The last thing that we have to deal with is environmental dissimilarity. If we look at a polluted environment and a clean environment, they can be different just because they're two different environments. It doesn't have to be related to the pollution. I mean, the microbes there can be different. So the way we deal with that is to sample across perturbations and across time, to look at the same environment and look at it through time or through changes in the environment. I'm going to give an example by looking at the effects of farming on the soil microbiome. And specifically, we're asking if we can find new microbes that would enable us to develop drought-resistant plants. And for this, we're using a very old experiment, a 50-year-old experiment, which is basically a giant lab plate. This is a giant lab plate, and it's a field. It has 4 replicates, 4 biological replicates of everything that we're doing. It has a side that is irrigated and a side that is rain-fed, and since it's the desert, the rain-fed side doesn't get a lot of rain. So it's really in deep drought. It has 2 phosphorus fertilization levels and 4 nitrogen fertilization levels. And it has 3 tillage treatments, which is how much you plow the land. Do you not plow at all, or do you plow very aggressively? And 2 rotation treatments, which means you either grow the wheat every year or let the soil rest a little bit. And it has 50 years of data, basically, and multiple drought spells each year. And in this data, we know what they used in the agronomy. We know what they used to plow the fields and so on in each one of these plots. We know the timing of wheat development. We know meteorology. There's a station there that studies the meteorology. And we know how much wheat grew and what's the protein content of the wheat and all of that. But we didn't have a microbiome data from this field. So we went out and collected it. And this is the same field, except before the season, so it's completely dry. And we managed to employ a barcoding system so we can locate the samples in the fridge afterwards. And we sampled all of the plots. This is me in a cowboy hat. We sieved them to remove all the insect and plant material, so we'll have just microbes in the soil. And then we flash froze it on site so we can analyze the DNA and RNA, and it will be fresh once we analyze it. We also bagged soil for chemical analysis so we can look at the chemistry of the soil behind it. And this was a great lab outing, but it also produced very, very good data. And I'm going to give you some preliminary results from this data. So first of all, this is a principal component analysis. Don't be scared. Each dot here is one of the plots. And if 2 dots are close together, it means they're similar. And one thing that we're seeing very clearly is that the dry plots, the ones that experience drought, have very different microbes, the ones on red, than the ones that are irrigated. So drought affects microbes. The second thing that we see from this plot is that in the dry, non-irrigated plots, other agricultural treatments also affect microbes. So we can see a difference between rotation, whether we let the ground rest, or no rotation, whether we grow wheat all year long. The second thing is that just by using the microbes from before the season, we can build a machine learning predictor that would predict how much wheat would grow at the end of the season. So on the y-axis is what we predicted, and on the x-axis is what actually grew. And you can see that there's a correlation between the amount of wheat that grew and what we predicted just by looking at the microbiome. The last thing is that we can dive into these microbes and look at their genome and find genes that are also predictive of how much wheat will grow. And this can help us to find genes that can develop new probiotics for wheat. And this could be a good and sustainable way, and also cost-effective, of developing probiotics for crops and helping places that have droughts year-round, like Africa or India. So to conclude, I hope I convinced you that that microbes are important, first of all, that by looking at these microbes and tackling the challenges that I showed you, we can use microbes for the benefit of humanity.